Good morning. In this video, I'll be discussing about metabolism of sulfur containing amino acids. The two major sulfur containing amino acids present in our body are methionine and cysteine. In this, methionine is an essential amino acid. That means it cannot be synthesized in our body and it has to be supplemented only through diet. Whereas cysteine is a non-essential amino acid, which means it can be synthesized in our body with the help of two other amino acids, methionine and serine. When you compare the structure of cysteine and serine, they differ in only, there is only one difference. What is the difference means? In cysteine, you have a sulfur atom, whereas in serine, instead you have an oxygen atom. So if you replace this oxygen atom with the sulfur atom, then serine can be easily converted to cysteine. Okay, so for this replacement, the sulfur atom is provided by methionine. Okay, so thus methionine and serine helps in the synthesis of cysteine. So now we will see how the methionine can release its sulfur atom for the synthesis of cysteine. We will discuss in detail. Okay, so when you see the structure of methionine, the sulfur atom is tightly locked between two methyl groups by a thioether linkage. So if you want to release this sulfur atom, first you have to remove this methyl group. For this, first what we are doing is we are attaching an adenosyl group to the sulfur atom of the methionine and converting it into S-adenosyl methionine. Okay, when an adenosyl group is attached to the sulfur atom of methionine, it becomes S-adenosyl methionine. The enzyme for this reaction is methionine adenosyl transferase, M shortly abbreviated as MAT, methionine adenosyl transferase. Okay, as the name says, it transfers an adenosyl group to the sulfur atom of methionine and converts it into S-adenosyl methionine. In this process, when an adenosyl group is attached to the sulfur atom, the bond between the methyl group and the sulfur atom will become weaker. So in result, what will happen means that subsequently this methyl group will be easily lost from it and the S-adenosyl methionine can be converted to S-adenosyl homocysteine. So this methyl group will be released. This, as you attach the adenosyl group, this bond becomes weaker and this methyl group will be automatically released in the next step by an enzyme called as methyl transferase. Okay, so this methyl group released in turn can be taken up by various substrates and it can be converted to methylated products. Okay, this process is called as transmethylation reactions, but about that we will be discussing little later. But as of now, you just know the methyl group will be removed from SAM and SAM will be converted to S-adenosyl homocysteine. Okay, now next, why we attach this adenosyl group? To remove this methyl group. We have removed the methyl group already. So the function of this adenosyl group is over. So in the next step, by an enzyme called as S-adenosyl homocysteinase, this adenosyl group will be chopped and S-adenosyl homocysteine will be converted to homocysteine. Okay, so far what we have done, we have removed the methyl group and we have exposed the sulfur atom. Now this sulfur atom can easily react with serine, can be easily donated to the serine by a transsulfuration reaction. Okay, so by transsulfuration reaction means what happens means sulfur atom from homocysteine will be transferred to serine. Okay, in exchange for an hydroxyl group, resulting in the formation of homoserine and cysteine. Okay, so homocysteine reacts with serine to form homoserine and cysteine. Okay, so the sulfyl group and the hydroxyl group will be exchanged between homocysteine and serine, so that we are getting homoserine and cysteine. Okay, so this is called as transsulfuration reaction. But this transsulfuration reaction will not happen directly. Initially what will happen means homocysteine and serine, they will condense with each other to form an intermediate called as cystathionine. Next, in the subsequent step, this cystathionine will be broken down to release homoserine and cysteine. Okay, so this is how cysteine is synthesized from methionine, from serine, with the help of methionine. Okay, so in this step, the first process, the condensation of homocysteine and serine to form cystathionine, is catalyzed by the enzyme cystathionine synthase, which is actually a vitamin B6 or pyridoxal phosphate dependent enzyme, whereas the breakdown of cystathionine to release cysteine and homoserine is catalyzed by the enzyme cystathionine lyase. Okay, so by the help of two enzymes, cystathionine synthase and cystathionine lyase, what is happening? Transsulfuration reaction happens and cysteine is synthesized. Okay, so after donating the sulfur atom to the cysteine, the remaining carbon skeleton, the homoserine, what they will do means they will undergo transamination process, they will undergo oxidative decarboxylation process, and ultimately it will be converted to propionyl CoA. This propionyl CoA formed in turn, they will enter the TCA cycle as succinyl CoA. Through succinyl CoA, it will be utilized for gluconeogenesis. Okay, that is why methionine is also called as glucogenic amino acid. Based on the catabolism, methionine is classified under glucogenic amino acid. Okay, so this is how methionine is catabolized, and during the same process, the sulfur atom is donated and cysteine is synthesized. Next, we will see how this cysteine undergoes catabolism in our body. So like many other amino acids, the first step in the catabolism of cysteine is removal of the amino group by a process called as transamination reaction. When you see the structure of cysteine, this cysteine is nothing but an alanine with an extra sulfhydryl group. Okay, so this is the cysteine. In this part is nothing but an alanine. So when an alanine with a sulfhydryl group is called as cysteine. Okay, so from this alanine, if you remove an amino group, what it will be forming? Alanine by transamination, it will be forming pyruvate. Okay, so cysteine, when it undergoes transamination, it will also be forming an alanine but with an extra sulfhydryl group. Okay, so that compound is nothing but beta mercaptopyruvate. So cysteine by transamination reaction, by the cysteine transaminase enzyme, the amino group will be removed from it and it will be forming beta mercaptopyruvate. Okay, next in the subsequent step, the sulfur atom will also be removed and it will be converted to pyruvic acid. And this pyruvic acid, through a reversal of glycolysis, it can be utilized for glucose synthesis. That is why cysteine is also considered as a glucogenic amino acid. Okay, so based on synthesis, methionine is an essential amino acid, whereas cysteine is a non-essential amino acid. But whereas based on catabolism, both methionine and cysteine both are glucogenic amino acids. Okay, now the sulfur atom released during this process will be removed from our body either as hydrogen sulfide or thiocyanate or as thiosulfates. Okay, so this is how cysteine is catabolized in our body. Apart from this, there is also one more pathway by which cysteine can be catabolized. Okay, so that pathway is also almost similar, but what is the one difference means, initially before undergoing transamination, the sulfur atom present in cysteine will be oxidized by an enzyme called as cysteine dioxygenase. Okay, so this cysteine dioxygenase will oxidize the sulfur atom and converts it into cysteine sulfinate. Okay, after this, the procedure follows the same like the previous pathway. Okay, so it will undergo transamination to form sulfinyl pyruvate, then it undergoes removal of sulfur atom and ultimately it will be forming pyruvate, which in turn will be utilized for gluconeogenesis. Okay, so in both the pathways, the destination is same, but the routes are different. Okay, so in first pathway, just transamination, after that removal of sulfur atom. Here initially oxidation takes place, which will be subsequently followed by transamination and removal of sulfur atom to ultimately form pyruvate. Okay, so this is how methionine and cysteine are synthesized and catabolized in our body. Now, before ending up, we'll be just discussing some of the disorders which are related to this anabolism and catabolism of sulfur containing amino acids. There are many different disorders which are related to the metabolism of sulfur containing amino acids, but the most important among them is homocystinuria. So what is homocystinuria means? Normally the serum homocysteine level is between 5 to 15 micromoles per liter. Normally in the blood it is between 5 to 15 micromoles per liter. At this normal level you cannot find any homocysteine in the urine. But when it increases beyond this, we call it as homocystinemia. When the blood homocysteine level increases beyond this normal range, we call it as homocystinemia. Okay, at that point the excess homocysteine can come out in the urine and it can lead to homocystinuria. Okay, so about this homocystinemia or homocystinuria, so what are the three things you should know means, what is the cause, what are the causes for homocystinemia, what are the effects it may produce, and finally how to diagnose it. Okay, so I'll be discussing about this homocystinuria under three headings. So first we'll start with causes of homocystinuria. So when you see the metabolism of sulfur containing amino acids, homocysteine is formed as an intermediate during the conversion of methionine to cysteine. So during the interconversion of methionine to cysteine, homocysteine is formed. This homocysteine form has two different fates in our body. One is it can be reconverted back to methionine or it can be converted to cysteine. So these are the two different fates of homocysteine. For converting the homocysteine to methionine, we need the enzyme methionine synthase, or otherwise called as homocysteine methyl transferase. Okay, and this enzyme, it utilizes folic acid and B12 as its coenzyme. Okay, so for the conversion of homocysteine to methionine, we need the enzyme methionine synthase and we need the coenzymes vitamin B12 and folic acid. On the other hand, for the conversion of homocysteine to cysteine, we need an enzyme called as cystathionine synthase. We need an enzyme called as cystathionine synthase, and this enzyme, it requires vitamin B6 as the coenzyme. Okay, but whenever any of these enzymes or coenzymes are defective, homocysteine cannot be converted to methionine and cysteine. So what will happen? They will be accumulating in the blood, leading to homocystinemia and homocystinuria. Okay, so from this itself you can easily make out what are the causes for various causes for the homocystinemia. Okay, so various causes are nothing but either the enzyme deficiency of the enzyme cystathionine synthase or methionine synthase. Okay, apart from this, there is also one more enzyme which is needed for producing this methyl tetrahydrofolate, which is nothing but N5N10 methylene tetrahydrofolate reductase. Okay, N5N10 methylene tetrahydrofolate reductase enzyme. Okay, so when any of these enzymes are deficient, it can lead to homocystinemia or homocystinuria. Apart from these enzyme deficiencies, even the coenzyme deficiencies like vitamin B6, B12 and folic acid deficiency can also lead to homocystinuria. And apart from these enzymes and coenzyme deficiencies, even hypothyroidism, old age, smoking and excess alcohol consumption can also lead to homocystinuria. Okay, so these are the various causes for excess homocysteine production. Now we will see what is the effect of this excess homocysteine production. So what is its major adverse effect means? It increases the risk of atherosclerosis. So why it increases the risk of atherosclerosis means by various mechanisms. It is not by a single mechanism. Through various mechanisms, the excess homocysteine can affect the blood vessels and it can lead to atherosclerosis. In that, the first mechanism is the excess homocysteine. They can interact with the lysine residues present in the collagen crosslinks within the blood vessel endothelium. Okay, so through this process, they can lead to endothelial damage and in turn can trigger the process of atherosclerosis. Okay, so this is one mechanism. Alternatively, what is the second mechanism means the excess homocysteines can lead to increased free radical productions, which in turn can oxidize the LDL. It can modify the LDL, and this modified LDL, they can be taken up by by macrophages through the scavenger receptors, which in turn can lead to foam cell formation, and ultimately it can end up with atherosclerosis. Okay, this is the second mechanism. Third mechanism is it also activates factor number 12. Hageman's factor will be activated. Okay, so once the clotting mechanism is activated, it leads to excess clotting, coagulation, and ultimately it can lead to atherosclerosis and blockage of the vessels. Okay, so by all these mechanisms, the excess homocysteine levels, it increases the risk of atherosclerosis, which in turn it increases the risk of myocardial infarction, stroke, and also peripheral vascular diseases. Okay, so this is one of the major complications we can expect in the patients with homocystinemia or homocystinuria. Apart from this, the patients with homocystinemia or homocystinuria, they can also present with Marfan-like features. Okay, then dislocation of lens can be there, then mental retardation and also neural tube defects are also common, especially in children who are born with congenital homocystinemia. Okay, so these are some of the complications or effects you can see in excess homocysteine patients with excess homocysteine levels. Okay, so now how to diagnose this means very simple, by measuring the blood or urine homocysteine levels. Okay, just now I told you in the blood the normal range is between 5 to 15 micromoles per liter. Okay, if it goes beyond that, then we can easily make the diagnosis as homocystinemia, and these patients are more prone for all these complications. Okay, so that's it about homocystinuria. Apart from homocystinuria, there is also one more condition called as cystinuria. The name itself says cystinuria means excess excretion of cysteine in the urine. Okay, what is cysteine means? It is nothing but the dimer formed by the conjugation of two cysteine molecules. When two cysteine molecules join with each other, they form cysteine. Okay, so this cysteine, when it will be formed, means normally in amino acids will never be seen in the urine. Why? Means though it is small in size, free amino acids, though it is completely filtered by the kidney, renal tubules will be completely reabsorbing it. Okay, so for reabsorbing each and individual amino acid, we have many different transporters in the renal tubules. For reabsorbing cysteine, we have a special transporter which along with cysteine will also reabsorb ornithine, arginine and also lysine. Okay, so when this transporter is defective, along with all the other amino acids, cysteine will also be excreted more in the urine and leading to cystinuria. Okay, so when this cysteine is excreted excess in urea, they will condense with each other to form a cysteine dimer, and that dimer in turn, because of the acidic pH of the urine, they will crystallize and they will lead to renal stone formation. They will, these patients, they mainly present with renal calculi. Okay, so how we can diagnose this means, usually the patients with excess excretion of cysteine or homocysteine in the urine, they will be showing positive response for cyanide nitroprusside test. Okay, so what is the treatment for these patients means, you can advise them to have plenty of fluids, and also you can give alkalinizer so that the pH of the urine will be increased and the chances for precipitation of cysteine will be reduced, so that stone formation can be reduced. Okay, so apart from stone formation, there won't be any much major complications in this patient. So if you can just prevent that stone formation with these measures, then this will not be producing any complications. Okay, next, apart from this, there are also few other diseases like cystinosis. So what is the cystinosis means? Excess accumulation of cysteine in the tissues, various tissues, due to the defective protein called as cystinosin, which is actually a lysosomal protein which is responsible for the export of cysteine from the lysosomes. Okay, so when this protein is defective, the cysteine amino acid can deposit in the tissues and it can lead to damage of the tissues. Okay, so this cysteine accumulation, it mainly happens in kidney, cornea, liver, spleen and WBCs. When it gets deposited in the kidney, it can cause damage to the renal tubules, which in turn can affect the reabsorption of various components, leading to a condition called as Fanconi syndrome. When it deposits in the cornea, it leads to photophobia. And when it deposits in liver, spleen and WBCs, especially in WBCs, that can be used as a diagnostic tool. Okay, so excess WBC, cysteine levels in the WBC can be used as a diagnostic marker for this condition, cystinosis. Okay, next, apart from this, there are also few other minor disorders like cystathionuria, which is caused by the deficiency of the enzyme cystathionase. Next, hypermethionemia, which is caused by the deficiency of the enzyme methionine adenosyl transferase, the first step, which is the first enzyme which is involved in the methionine catabolism. When that is defective, methionine cannot be catabolized in our body, so it will accumulate, leading to hypermethionemia. Okay, next, the third disorder, Host-House syndrome. Here, the reabsorption and absorption of methionine from diet, from the intestine and kidney will be defective, leading to methionine deficiency. Okay, so these are some of the major disorders that are related to metabolism of sulfur containing amino acids. Okay, so to summarize, the major disorders are homocystinuria, cystinuria, cystinosis, cystathionuria, hypermethionemia, Host-House syndrome. Okay, for all these things, you should know what is the major biochemical defect. For homocystinuria, it can be either due to the enzyme deficiencies or coenzyme deficiencies which are required for converting homocysteine to cysteine or methionine. Okay, the major enzyme deficiencies are cystathionin synthase enzyme deficiency, methionine synthase enzyme deficiency, or N5-N10 methylene tetrahydrofolate reductase enzyme deficiency. Whereas coenzyme deficiencies, vitamin B6, B9 and B12 deficiencies. Okay, all these can lead to homocystinemia and homocystinuria. Next, cystinuria is due to the defective renal transporter which helps in the reabsorption of the amino acids like cysteine, ornithine, arginine and lysine. Next, cystinosis is due to the defective protein cystinosin. Then cystathionuria due to the enzyme deficiency cystathionase, hypermethionemia due to methionine adenosyl transferase enzyme, and Host-House syndrome due to the defective transport of methionine in the intestine and in the kidney. Okay, so that's it about the metabolism of sulfur containing amino acids. Thank you.